Thanks a lot, Josh, for that nice introduction, and thanks everyone for coming. So my lab is largely interested in understanding the genetic etiology of brain disorders with the focus of genome-wide association studies, or GWAS. And GWAS is the study of identifying variants, and the overarching goal of my lab is to link these variants to specific function. And again, my lab is largely interested in brain disorders or psychiatric disorders. So the overarching goal is to link variants to the brain function. And we and others often think that the first step in linking variants to function is to identify the target genes of the variants, with the hypothesis that once we identify the genes, it's relatively easier for us to understand the biological pathways, cell types, and circuits that are involved with a given trait, which could potentially provide us some mechanistic understanding. And this is largely based on the very well-depth studies on the gene function in terms of biological pathways, as well as also rich information about which cell types or which circuitry that these genes are highly expressed of. So in the first part of my talk, I'm mainly going to talk about how we would like to map variants to the target genes using substance use as a use case. And in the second part of my talk, I will go to the very first part of the diagram to talk about our recent work on how we would like to prioritize the variants from the very beginning using schizophrenia as a use case. So with that, let's focus on the first question. And this really became the central aspect of my lab, largely thanks to my graduate student, Nancy, who has a picture here. So she's my first graduate student. And when she came to my lab, she told me that she's interested in addiction biology or genetic etiology of substance use. And I was very puzzled when she told me that, because my lab, or I have never really worked on addiction at all before. So I asked her that I'm not really working on that. Are you really sure that you would really like to do this and you would still like to join my lab? And her answer was yes. And she made a really great stride since then. And then she really made the substance use genetics as one part of the lab. So because of her, I was also attracted to this study so that I can study the genetic etiology of substance use. And what was really surprising to me was that substance use phenotypes really give a great platform to us to study gene environment interactions. And another important aspect of substance use genetics is that depending on which substances that you're using, either alcohol or cigarettes, you can actually get the different genetic makeup. Let me use my mouse because there are some people in the Zoom too. So for example, if you are measuring the genetic architecture of alcohol use, it's actually not necessarily the same as the genetic architecture of the cigarette consumption. And another very interesting aspect is that different behavior is encoded in the genome in a different manner. So what I mean by that is that for some of the substances like alcohol, we all drink alcohol, but it does not necessarily mean that we're all alcoholic. And that's actually encoded in our genome. So if you measure the genetic correlation between alcohol consumption and alcohol dependence, the genetic correlation is not perfect. You roughly get 0.6 of genetic correlation between consumption and dependence measure. However, for some of the substances that are way more addictive, like cigarettes, consumption and dependence are actually encoding almost the same thing. So that the genetic correlation between cigarette consumption and cigarette dependence or nicotine dependence is higher than 0.9, meaning that cigarette consumption can be a really good proxy in understanding the nicotine dependence. So to summarize almost the decade-old study of what we have learned from substance use genetics, we now know that the genetic contribution to substance use is substantial. And we have found out that out of many types of genetic variants, common variations seem to play an important role. And finally, there's not a single genetic variant that can explain the whole substance use biology, but hundreds of genetic variants can be associated with substance use. However, this is not really the end of the story and actually opened another question. And the major question here is that once we identify the variants, how we can actually infer the functional consequences of these variants. And this is a very central question in the field, largely because the majority of the variants that we identify from these GWAS are coming from the non-coding region of the genome that we do not really know how to interpret very well. And my lab has been actually built upon the three-dimensional chromatin structure. And because of that, we had a hypothesis that these risk variants that are associated with any trait can potentially affect the genes that are pretty distal away by forming these chromatin loops, so that the risk variant may not really affect the nearby genes, but can affect the genes that are distal if they are in close physical proximity in the three-dimensional space. So with that hypothesis, we have built what we call as tightly coupled magma, or H-MAGMA. And what H-MAGMA does is that it first groups the SNPs into two groups. The first group is exonic and promoter SNPs, and the second group is non-coding SNPs. And for those that are located in either exons or promoters, we directly map them onto the target genes based on where they're located. However, still the majority of the SNPs are unannotated, and for those SNPs, we use Hi-C interactions or chromatin conformation data sets to identify their target genes based on these three-dimensional landscape. And after that, we combine all this information together to generate what we call a SNP to gene level relationship matrix. And in this matrix, we can basically infer which SNPs can be mapped to which genes. So the overarching concept of H-MAGMA is that once you identify the gene to SNP level relationship matrix, you can go back to the GWAS summary statistics, which provides you the SNP level p-values. And then you can aggregate all these p-values per SNP into the gene level based on this relationship. So all these p-values for these 5 SNPs can be mapped onto this blue genes, and all of these 3 p-values from 3 SNPs can be also mapped to this yellow gene and vice versa. And using a tool that has been developed called MAGMA that can actually aggregate the SNP level p-value to the gene level, we can convert the SNP level p-value to the gene level p-value so that we can now see which genes are significantly associated with a given trait. So using this schematic, we try to leverage this framework to 4 GWAS that I mentioned earlier. So 4 related to cigarette smoking, 1 for cigarette consumption, and 1 for nicotine dependence, and another 2 GWAS for alcohol use traits, 1 for alcohol consumption, and the other one is alcohol use disorder. And then we try to basically generate the gene lists that are associated with substance use phenotypes. However, there's still an open question, which is largely from which Hi-C data set shall we actually infer this gene to SNP level relationship matrix? And this is largely because this relationship can be quite cell type and tissue specific. So we have to basically query this information from the relevant cell type or relevant tissue type. So this led us to go back to the circuit level mechanisms, because circuit level mechanisms for addiction has been really relatively well understood. So from the circuit level studies, we now know that there are mainly 2 brain regions. There are also, of course, other brain regions as well, but 2 brain regions are very central. And those brain regions include midbrains that largely contain dopaminergic neurons, which project to the prefrontal cortex, which also contain different cell types that could potentially play a very critical role in developing addiction. So we first wanted to see whether the circuit level mechanism could be translated back to the genetic level mechanism. And for doing that, we have run heritability enrichment analysis for the enhancers that are either located in the midbrain or the prefrontal cortex, and checked whether those enhancers are more likely to be enriched with genetic risk factors for 4 GWAS traits that we have measured. So simply, long story short, we have found that both enhancers in the prefrontal cortex as well as midbrain showed high heritability enrichment for all 4 traits, again showing that circuit level mechanism could be indeed translated back to the genetic level mechanism. And because both of these brain regions contain various cell types, we wanted to see which cell types seem to be most important. So it's been really well known that within the midbrain, dopaminergic neurons are central. So we wanted to see whether dopaminergic neuronal enhancers could potentially provide or explain the heritability enrichment that we have observed. And indeed, we found that dopaminergic neuronal enhancers were enriched for all these 4 GWAS traits. Cortex gave a little more complicated signal because of the cellular heterogeneity in the cortex. And out of 4 cell types that we measured, we found that neurons seem to be the central player in understanding heritability enrichment, although there seem to be some glial roles as well. So based on these results, we basically determined that there will be 2 neuronal subtypes that are implicated for substance use disorders. And those are midbrain dopaminergic neurons, as well as cortical neurons generated or obtained from the dorsolateral prefrontal cortex. So using the Hi-C datasets from these two Hi-C datasets, we tried to generate this gene to SNP level relationship matrix so that we can infer the gene level p-values or significantly associated genes for each trait. So at the end of the day, we were able to identify roughly hundreds of genes that were associated with individual traits. However, you will be able to acknowledge that nicotine dependence is an outlier here, that it only identified dozens of genes. And this is not surprising because nicotine dependence was the least powered GWAS among 4 GWAS that we have identified. And because of that, oftentimes in the following work, you will be able to see that nicotine dependence acts often as an outlier. And we don't think that it's actually showing different biology, but it could be largely coming from the low power GWAS. So going back to the original diagram, I told you earlier that once you identify the genes, it's relatively easier for us to understand the biological pathways, cell types, or circuits that are involved with the disorder. So I'm going to show you how identifying these genes from this data set could potentially provide us some information about substance use biology. So first, we wanted to check which biological processes are associated with substance use. And as a proof of principle concept, and somewhat reassuringly, we found some very clear evidence that we are finding something right. So for example, problematic alcohol use and drinks per week were not only associated with other terms, but the major term involved ethanol metabolic process as well as response to alcohol. This is pretty much expected because we would expect that ethanol metabolism is a key process when we are measuring genetic etiology of ethanol use. Similarly, nicotine dependence and cigarettes per day were associated with cholinergic and nicotinic pathways, which are again pretty much reassuring because we all know that cigarette is actually provoking those systems. However, we found other interesting pathways involved in these 4 traits, and those include neuronal death or apoptotic pathways that, oh, I lost my cursor now. Where is it? Oh, sorry. I still lost it. Sorry, let me just use this. Yeah, so first of all, I found that neuronal apoptosis was also associated, but it was not only neuronal apoptosis, but I found some neurodegenerative pathways. So tau protein binding was associated with drinks per week, and amyloid beta metabolism was also associated with cigarettes per day. And in addition to that, I found that pain perception was associated with drinks per week and cigarettes per day. And that was also somewhat expected because we all know that opioid crisis has been actually provoked by pain prescription. So we thought that addiction has to be associated with pain prescription. And we also found that. And finally, we found neuron oligodendrocyte crosstalks. And this was pretty interesting because all of these gene lists have been already queried from dopaminergic and cortical neurons. So we thought that we would see a lot of neuronal related pathways, but even compared to the neuronal related pathways, we also found that oligodendrocytes seem to play a role. So this suggests that there might be some neuron glia crosstalks that are involved with substance use genetic etiology. Secondly, we wanted to see what kind of cell types are associated in explaining substance use risk. And using cortical neuronal H-MAGMA, we wanted to see which cortical neurons are playing an important role in understanding substance use biology. And here we found that, oh, again, I can't find it, sorry. I found that upper layer excitatory neurons seem to play a role. And in addition to that, these inhibitory neuron subtype 6, which stands for parvalbumin inhibitory neurons, seem to be shared among problematic alcohol use, drinks per week, and cigarettes per day. And again, you can see that nicotine dependence was an outlier, and this is not really necessarily suggesting that nicotine dependence is really an inhibitory neuronal problem, but I think it's largely because of the low power GWAS of nicotine dependence. And in addition to that, when it comes to dopaminergic neuronal H-MAGMA, we found that it was not the fully mature dopaminergic neurons that seemed to be most enriched with, but we found that intermediate dopaminergic neurons across dopaminergic neuronal differentiation seem to express these genes most highly of, suggesting that dopaminergic neuronal development can potentially predispose an individual to be more susceptible to substance use. We finally wanted to see whether our genetic level mechanism can be now fed back to the circuit level mechanism. So in doing that, we wanted to see whether the gene list that we have we have largely obtained from cortex and the midbrain can be actually used to infer other addiction-related brain pathways that could give us some information about circuit level mechanism. And interestingly, we found that in addition to cortex and the midbrain, we found that amygdala and thalamus were also showing high expression of these genes, again suggesting that even though we're not really getting the Hi-C data sets or gene list from all different brain regions, we could potentially still use the genetic risk factors or the gene information from certain brain regions and can actually expand that to other brain regions to infer some circuit level mechanisms. And then finally, we wanted to see whether this level of information could be expanded to other types of substances. And this is largely because we were only testing 2 substances here, cigarettes and alcohol, while there are so many different substances that are affecting human health. And especially in the US, opioid use has been a really central problem. However, we do not really have a really well-powered GWAS when it comes to opioid addiction. So we thought that maybe we could leverage the genetic information that we have obtained from cigarette smoking and alcohol use behavior so that we can learn something about opioid or other types of substance use until we get a better powered GWAS. So actually supporting this idea, we found some extensive crosstalks between substance use phenotypes. So for example, problematic alcohol use was not only associated with ethanol metabolism, but we found that it was also morphine and nicotinic response that seemed to be involved. And similarly, when it comes to nicotine dependence and cigarettes per day, again, not only cholinergic and nicotinic pathways were involved, but alcohol catabolism was associated with nicotine dependence and cocaine response was associated with cigarettes per day. So this suggested that there seems to be some genetic etiology shared across different substance use genetics. So with that idea, we wanted to see whether we could identify the pathways that are shared between cigarette smoking and alcohol use behavior, and whether that could infer some generalizable addiction mechanisms. And supporting this idea, when we compare genetic correlation between problematic alcohol use and cigarettes per day, while the genetic correlation was not super high, we found some meaningful genetic correlation. So because of this, we performed rank-rank hypergeometric overlap, or RHO. And what it does is that it ranks the gene list for problematic alcohol use based on the H-MAGMA output, and also rank the gene list for cigarettes per day based on H-MAGMA output, so that any genes with significant p-values will be ranked more top. And then you could actually infer the genes that are shared in both of these lists. And by running this, we were able to find some difference between dopaminergic neurons and cortical neurons for the first time. And what we have observed from here was that compared to cortical neuronal H-MAGMA, then we identified only 120 genes, dopaminergic neuronal H-MAGMA seemed to identify roughly 3,000 genes to be shared between cigarettes per day and problematic alcohol use, which potentially suggest that there might be some circuit-specific mechanism in understanding pleiotropy. And potentially that pleiotropic mechanism can largely arise from midbrain dopaminergic neurons than cortical neurons. So we named these gene sets that have been shared between problematic alcohol use and cigarettes per day as pleiotropic genes, and we wanted to know what those genes were doing. So first, we wanted to see what kind of biological pathways were involved with pleiotropic genes, and we found that they were largely neuronal or synaptic involved, which is not surprising at all because we found actually these gene lists based on the neuronal Hi-C data sets. However, when we checked their cellular expression levels within the midbrain, we found that it was not only dopaminergic neurons that showed high expression of these genes, but we also found that midbrain GABAergic neurons and serotonergic neurons were also involved, suggesting that it is not only just one cell type within the midbrain, but multiple neuronal subtypes within the midbrain can all collegially affect the pleiotropic mechanism. And in addition to that, again, across dopaminergic neuronal differentiation, we found that DA1, or the intermediate dopaminergic neuron, seemed to be central for understanding pleiotropy. We finally wanted to see whether these pleiotropic genes indeed play some important roles when it comes to other types of substances that we haven't even tested. And in this case, we used cocaine treatment. So in this study, they have dissected nucleus accumbens from the cocaine-treated mice and then performed single-cell RNA sequencing. And out of many different cell types within the nucleus accumbens, they found the largest differential expression signal that has been elicited by cocaine in these cell types that are involving dopaminergic receptor expressing medial spiny neurons. So we compared our pleiotropic genes with cocaine-elicited DEGs and found that there were significant overlap. And in addition to that, even though these genes were not significantly always differentially regulated, when we compare the gene expression of all these 3,000 genes, we found that they were pretty much consistently downregulated upon treatment of cocaine. So suggesting that even if we generated this gene list from two totally different types of substances, maybe the mechanism that we identify from these gene lists could potentially inform us when it comes to the mechanism of other types of substance of abuse. So to summarize the first part of my talk, I hope that I convinced you today that we can potentially use H-MAGMA to map variants to the target genes. And by mapping GWAS for substance use, we were able to identify biological pathways, cell types, and circuits that are involved with substance use biology. So with that, I am going to the next part of my talk, which is how we would like to prioritize the variants. And the reason behind this is because we finally came to the point where we have too many variants, too many genes problem. So for example, when we apply H-MAGMA to schizophrenia GWAS that has been published in 2018, we identify 5,000 genes to be implicated in schizophrenia. And even if I generated this gene list, I don't really believe that all of those genes will be causally implicated in schizophrenia. So there's one way to actually prune down these gene lists, and that's by just focusing on genome-wide significant loci. So H-MAGMA basically leverages genome-wide SNP level information. So it still converts the SNP level p-values when it's underneath this genome-wide significant threshold. However, by just focusing on genome-wide significant loci, you're only looking at these loci that pass the genome-wide significant threshold. And from these loci, you can actually even further narrow down the variance by performing computational fine mapping algorithm that gives you what we call as credible SNP list, that gives you basically the smallest set of SNPs that are that can explain individual locus. And by leveraging multiple gene mapping algorithms, we can actually map these variants to roughly 1,100 genes. And even if this is 1/5 of the original gene list, we still think that this is too many. So we were wondering whether there is a way that we can even further prioritize the variants coming from these fine mapped schizophrenia variants. So for doing that, we have performed massively parallel reporter assay, or I will call it MPRA from here. And this has been largely spearheaded by two very talented graduate students, Jessica and Sol. So here, what we have done was that we put schizophrenia risk variants. So all those 6,000 schizophrenia risk variants that I mentioned earlier, an individual variant is going to have 2 alleles. So we were roughly testing 12,000 alleles here in the same assay. And we put them in front of the promoter and the GFP, which is a reporter gene, followed by the 20 nucleotide barcodes. And then we packaged this into the AAV, which was then transduced to human neuroprogenitor cells. And within human neuroprogenitor cells, what we would expect is that risk allele and protective allele for a given variant is going to affect the gene expression of the GFP. And this expression elicited by these genetic variants can be measured by performing RNA sequencing on the barcode. And we already know which barcodes are pre-mapped to the variant. So just by quantifying the barcode expression level, we can infer which variants or which allele can it be actually mapped back to. So here, the major hypothesis is that if a given variant has different barcode expression level between risk and protective alleles, we can call them as regulatory variants or MPRA-positive variants. And we would like to distinguish these MPRA-positive variants from MPRA-negative variants that do not show such allelic regulatory activity. And here I'm not calling this as regulatory or non-regulatory variants because I don't really expect that all MPRA-negative variants are going to be truly non-regulatory because some of them can affect alternative splicing or other types of gene regulatory mechanisms that we are not really testing here. So with that, we implemented MPRA on 6,000 variants that I mentioned earlier. And out of those 6,000 variants, some of the variants were not really testable using MPRA, largely because they overlap with the molecular cloning strategies that we are using, as well as some of the variants were actually missed by barcode dispersion during the cloning process. So at the end of the day, we were able to test the regulatory activity of 51,000 variants. And out of these 51,000 variants, only roughly 400 variants showed regulatory activity. If you see the volcano plot of these variant function, you can acknowledge that we are not only identifying the significant variants, but we can also find the directionality of the effects of these variants, meaning that we now know whether the risk allele, that is basically increasing the risk for developing schizophrenia, is increasing their gene expression or decreasing their gene expression. And on top of that, we're not only narrowing down the variant list from just 5,000 to 400, but we can actually dramatically reduce the number of SNPs per locus, meaning that even after performing fine map, still a huge chunk of the loci could be explained by more than 50 variants, but by performing MPRA, we can usually explain a given locus by less than 5 variants. And this is giving a perfect example of how MPRA can greatly narrow down the SNP list. So this GWAS locus, as you can see, has a pretty complex signal, and it's located in CAPNA1C gene. And out of these complex genetic signal that is elicited by LD, you can actually narrow down this region into roughly a little more than a dozen SNPs using fine mapping algorithm. However, once you perform MPRA, you can still narrow down this like a little more than a dozen SNPs into a single variant that has the allelic regulatory activity. So this case, it gives you a perfect example of how MPRA can greatly refine the SNP. And once we map this SNP into using three-dimensional chromatin structure, we found that this SNP was actually interacting with one of the promoters of CAPNA1C. So since we now have the MPRA-positive variants that we can follow up, we wanted to see whether we could use this data set to basically train and infer what is the fundamental regulatory logic of the variants that we identified. And in other words, we basically wanted to find what are the distinguishing factors for MPRA-positive SNPs. And the major hypothesis behind this is that if we actually really identify the regulatory logic to predict MPRA-positive SNPs, we can potentially use this information and expand it to other GWAS that we haven't really tested experimentally. And then we can just infer which variants are causal or which variants are regulatory versus not, just based on the MPRA data set that we got. So we have compiled multiple hypotheses on what makes a causal variance. And we tested whether those hypotheses actually can distinguish MPRA-positive variants from negative variants. So first, we wanted to see whether the variants with strongest association statistics coming from GWAS are likely causal. Secondly, we tested whether variants with higher posterior probability from the fine mapping algorithms are likely causal. Thirdly, we wanted to see whether evolutionary conservation matters in a sense that evolutionary conserved variants are more likely causal. And finally, I think this is the most prevailing hypothesis in the field, that variants within enhancers or variants within chromatin accessibility or variants that change the transcription factor motifs can potentially play a causal role. So we wanted to test an individual hypothesis. So first, we wanted to check whether GWAS summary statistics differ between MPRA-positive and negative variants, and the simple answer was no. When we compared GWAS p-value or GWAS odds ratio, we could not really find some clear distinguishment between MPRA-positive and negative variants. And in addition to that, out of 102 loci that we identified MPRA-positive hits, only 12 loci contained MPRA-positive variants that also showed regulatory activity. So this means that if you're just following up on index SNPs, which basically means the SNPs with strongest association at a given locus, you will probably be wrong in more than 90% of the cases. Secondly, we wanted to see whether fine map posterior probability differs between MPRA-positive and negative variants. And again, simple answer was no, that fine mapping algorithm can potentially narrow down the SNP list, but posterior probability itself does not provide any meaningful information about regulatory information. And in addition to that, there was also no difference in evolutionary conservation between MPRA-positive and negative variants. And this was calculated and inferred recently from Zoonomia Consortium by comparing roughly 240 mammalian species genome. We finally checked whether functional annotation can potentially explain the difference between MPRA-positive and negative variants. And the very simple answer was no. Enhancer overlap alone did not really seem to distinguish MPRA-positive variants from negative variants. However, there seemed to be some cell type specific effects. So for example, MPRA-positive SNPs seem to be more located in neuronal enhancers compared to the rest of the glial enhancers. And on top of that, we found that early excitatory neuronal enhancers seem to have a higher overlap with MPRA-positive variants compared to the MPRA-negative variants. And we found the same thing from enhancers from astrocytes and oligodendrocytes in the fetal brain. However, in general, enhancer overlap alone did not really seem to really distinguish MPRA-positive from MPRA-negative variants. This was very, very puzzling to us because it is really well known that schizophrenia is a neuronal disorder. And people have found over and over that GWAS of schizophrenia is associated or enriched in neuronal or brain enhancers, but we're not really finding this. So we wanted to see whether these MPRA-positive variants are potentially more enriched compared to the background. So we leveraged two genomic backgrounds. The first one is the LD SNP, so those are local backgrounds. That means that we basically identified all the SNPs within the LD of this genome-wide significant loci, and then try to just identify the SNPs within those loci that are not associated with schizophrenia. And then we also generated the random background SNP set. So these SNPs are spread across the genome, so they are not located in schizophrenia genome-wide significant loci. However, they are matched with MPRA-positive variants when it comes to minor allele frequency as well as the LD structures. So even when compared to LD to MPRA-positive SNPs, we did not really see very significant enrichment of MPRA-positive SNPs within these enhancers. However, we started finding very significant enrichment for MPRA-positive SNPs when we compare them to the random background. So according to this study, what we are thinking is that these genome-wide significant loci are already showing elevated signal to neuronal enhancers or brain enhancers. And because they already put these loci into the elevated enrichment, it is very difficult to parse out the function within those loci just based on the enhancer overlap. So that was our takeaway from this. And finally, we wanted to see whether transcription factor binding occupancy can potentially parse out the MPRA-positive variants from negative variants. And when doing this, we did a little more complicated stuff than just the position-weighted matrix data set. So we tried to infer the transcription factor activity rather than just transcription factor binding by combining transcription factor binding with expression and transcription factor function. And when it comes to transcription factor binding, we did not only identify the position-weighted matrix, but when we are measuring whether the risk allele can disrupt or increase transcription factor binding, we have leveraged these data sets that have been generated from Bing Ren's lab that has actually experimentally tested the binding occupancy of the transcription factor and compared the transcription factor binding occupancy between risk and protective alleles. And they did it for roughly 400 transcription factors. And out of 400 transcription factors that they experimentally tested, they were able to generate this support vector machine only from roughly 90 transcription factors. So it means that they could predict the transcription factor binding activity based on the training data set that they have experimentally validated. And we only use those rather than using position-weighted matrix because whatever you do with position-weighted matrix, we never really found any relationship. And I think it's largely because it is very noisy. And because of this transcription factor binding occupancy is cell type agnostic, we try to incorporate cell type information by incorporating how much of these transcription factors are expressed in the cell types that we introduce these variants, so HMPs or human neuroprogenitor cells. And on top of that, transcription factor is not always an activator, but it can also act as a repressor. So when the transcription factor is a repressor, if the risk allele disrupts its binding, it means that the gene expression should be actually increased rather than decreased. So we also try to leverage that information. So once we actually taken into multiple aspects of transcription factor activity, we started getting some difference between MPRA-positive and negative variants. So that you can see that MPRA log fold change had some weak correlation with transcription factor activity that we predicted. And that relationship was totally abolished when we compare that with MPRA negative variants. And we wish that this relationship will grow once we have a better understanding of transcription factor. So if we can actually get this transcription factor binding occupancy for not only 94 transcription factors, but to 2,000 transcription factors that exist in the human genome, as well as if we know whether transcription factors are actually acting as a repressor or activator in human neuroprogenitors, because many of them can actually act both, I think this relationship will definitely grow. So going back to the very original part, I have shown you earlier that we were largely using Hi-C data sets to map variants to the target genes. However, actually in the field, the gold standard principle in mapping variants to the target gene is to utilize expression quantitative trait loci, or EQTL. So we wanted to compare our MPRA results with EQTLs to see how many of these MPRA positive variants can be mapped back to the EQTL data sets. And out of 9% of the variants that we found regulatory activity, we found only 36% of those variants were overlapping with brain EQTLs, while 64% of them did not show any EQTL evidence. And out of those that overlap with EQTL, reassuringly, we found that more than 80% of the variants showed identical direction of the effects with EQTL, showing that MPRA and EQTL are both suggesting the same direction of the effects when it comes to the gene expression. And out of those EQTLs that showed identical direction of the effects, again, more than 80% of those variants were also showing colocalization signals with GWAS, suggesting that these EQTL and GWAS overlap is not just spurious association coming from LD. So even though these parts were somewhat reassuring, we were pretty much puzzled by this graph showing that a lot of these MPRA positive variants do not show EQTL evidence. And this let us go back to a recent preprint coming from Jonathan Pritchard's lab that has shown that GWAS hits and EQTLs are identifying actually different set of variants. And in this study, they have shown that GWAS are more likely to affect or identify the variants that are distal to transcription start sites, and therefore they are more regulatory or distal enhancers-like compared to the EQTL identified variants. So with this, we wanted to see whether our MPRA positive variants that show no EQTL evidence, so this sets, versus MPRA positive variants that do overlap with EQTL and also show identical direction of the effect, so these variants, and wanted to compare whether those two sets of variants are indeed different. So when we first compare the transcription start site distance between MPRA positive variants that are non-overlapping with EQTLs and overlapping with EQTLs, we indeed found that MPRA positive variants that are non-overlapping with EQTLs were more distal to transcription start sites. And supporting that, we found that those variants that are not overlapping with EQTLs were more likely to be located in distal neuronal enhancers compared to the MPRA positive variants that were overlapping with EQTLs. So this made us go back to the original idea of how to map variants to the target genes, that maybe given that a lot of these MPRA positive variants are located in the distal regulatory elements that are not necessarily overlapping with EQTLs, we can potentially leverage Hi-C driven information in linking variants to target genes. So to do that, we have leveraged the long-range chromatin interaction data sets across human neuronal differentiation. So Hi-C data sets obtained from germinal zone and cortical plates coming from the fetal brain, as well as pediatric neurons and adult neurons. And then we compiled all the long-range interactome across these human neuronal differentiation so that we can map MPRA-positive SNPs to their target genes. And when we did that, again, very surprisingly, EQTL and Hi-C seem to identify largely non-overlapping set of genes. And we then wanted to go back to Jonathan Pritchard's preprint to see whether these GWAS hits are indeed identifying different genes, not only different set of variants, but also different sets of genes compared to the EQTLs. So in their studies, they have shown that GWAS hits are more likely to affect the genes with higher selective constraints. So if those genes are mutated, humans are more likely to be lethal. And also genes that have higher regulatory complexity. So those genes are more likely to have multiple enhancers and more complex enhancers. And finally, they have shown that GWAS hits are more likely to affect genes with functional annotations. So we wanted to test individual hypotheses. So when we compared the selective constraints between EQTL-guided genes and Hi-C-guided genes, we found that Hi-C-guided genes had lower LUF scores compared to the EQTL-guided genes. And this, if a given gene has lower LUF score, it means that that gene has basically is more susceptible or sensitive to any gene dosage effects. So if that gene has any mutations, people are more likely to become lethal. So this suggests that indeed Hi-C guided genes seem to have higher selective constraints. And secondly, we wanted to see how many loops were anchored to EQTL-guided genes versus Hi-C-guided genes. And again, we found that Hi-C-guided genes had higher number of loops compared to EQTL-guided genes. And finally, when we compared the gene ontology between Hi-C-guided genes and EQTL-guided genes, I know that the font is very small, but I think you will be able to acknowledge that there were way more gene ontologies that were involved with Hi-C based mapping compared to the EQTL-based mapping. So this really suggests that when it comes to identifying variant to gene relationship from MPRA-positive variants, we can potentially utilize Hi-C data sets than the EQTL data sets. So in mapping or integrating these two types of data sets together, we were wondering whether we could utilize another third sets of data. And that was largely because MPRA is all conducted in an episomal manner, which means that all of these plasmids or libraries are introduced into the cells without any chromatin. So these plasmids are just there in a nascent format. So we can actually test the potential variant effects when there's no chromatin at all, but still it means that we're not really testing the variant effects under the chromatin or endogenous context. So we thought that maybe we can borrow that endogenous context from a taxic data set so that while a taxic can provide us the endogenous chromatin context from the chromatin accessibility data, we can potentially use the Hi-C to infer these variant impacts onto the target genes. So for that purpose, we have proposed an accessibility by contact model, and this is actually building up on Jesse Engrid's activity by contact model, but he was basically using that to infer enhancer gene relationship. And here we would like to use this to infer the variant level activity onto the gene level. So for a given locus, what we are trying to do is when we're we can have the allelic activity that is measured by MPRA log fold change, and then we can leverage the chromatin context as well as the chromatin contact frequency so that we can infer its impact on the gene expression. So we simply weighted the MPRA measured allelic activity with chromatin accessibility in which these variant is located in. And we also counted the chromatin contact between this variant and the target gene that we would like to test. And then after that, we summed up all those expression within a given locus so that we can predict the gene expression. So if we say that target gene is predicted to be upregulated in a disease condition, we can actually then compare this against schizophrenic postmortem brain expression data set to see whether those genes are indeed upregulated in schizophrenia postmortem brain expression. So we first wanted to see how this model acts when we are only counting MPRA level activity. So only if we are testing allelic regulatory activity, what is the predictive power? And we found that when we were just summing up the allelic activity that was measured by MPRA, roughly 55% of the genes seem to have consistent direction of the effects with postmortem expression data. When we are putting chromatin contact information on top of that, we found that the predictability grew a little bit, that we now have roughly 57% of the genes with consistent direction of the effects. And finally, when we were including chromatin accessibility on top of that, we found that more than 60% of the genes showed consistent direction of the effects with schizophrenia postmortem brain expression. And this was also very statistically significant compared to the permuted distribution. So with that, to summarize the second part of my talk, I hope that I convinced you today that MPRA can provide a great avenue to study the variant effects, and we can prioritize the variants from GWAS using this high throughput screening system. And oftentimes the biological principles that we are actually gathering from MPRA or experimental validation can be distinct from the pre-existing hypothesis. And also, in mapping these variants to the target genes, potentially chromatin architecture, including Hi-C and ataxi, can provide some complementary system to link variants to the target genes, and also can complement the episomal nature of MPRA. So because we are very much intrigued by the results that we obtained from schizophrenia risk variants, I know that Alan and Josh are also within this consortium, but we are trying to basically expand this approach to roughly 250,000 variants associated with various human diseases. And we are going to introduce this to mouse circulation so that we can test the variant effects in multiple organs. And because many of these disorders and variants have sex-specific effects, we would like to test the variant effects in 2 different sexes of the mice. And finally, we're going to introduce half of these mice with immune stimulation so that we can see how these variants are affected based on the immune stimulation or some level of gene environment interactions. So with that, I would like to give a special shout out to especially these 3 trainees, Nancy, who really spearheaded the first work, and Jessica and Sol, who really spearheaded the second part of the work, and all my collaborators across UNC, Mount Sinai, RTI, and UCLA. And thank you for your attention. Yes. Oh.
Oh, thank you for the great talk. So I was really surprised by like H-MAGMA and EQTLs identifying different variants. But then with H-MAGMA assigning variants and promoters and exons to their closest gene, do you think that you might be missing some do you think that they might actually be acting as enhancers distal to other genes instead?
I think H-MAGMA will likely going to pin that interactions, right? Any distal relationships will be queried by H-MAGMA.
Oh, sorry, variants in promoters.
Oh, in promoters. Yeah, but H-MAGMA actually assigns promoter variant to that gene too. So we hope that those proximal interactions may largely also be retained because all the promoter variants are assigned to that gene. Is that what you're asking?
Oh, yeah, no, I guess because yeah, you know, there's some promoters that have some like that show enhancer-like behavior, like very small percentage, right? But you know, so
yeah, you can actually test that too. So in our study, we did not really do that. So we did not really map promoter variants to distal genes, largely because we think those variants are more likely to affect that gene rather than the distal genes. But you're totally right that when you are measuring the promoter anchored interaction, roughly 20% of them are connected to different promoters. What we are thinking is that Hi-C resolution is actually 10 kilobase, and our promoter definition here is 2 kilobase, right? So if the variant is indeed in close LD, there will be some variants that are very close to promoters, but are not really assigned as that promoter. And those variants should be still mapped to the other promoters. So we hope that it's not really going to miss too much information.
Yeah, that makes a lot of sense. Thank you.
Mhm.
Great talk. I have another question about H-MAGMA. So we've explored, connecting regulatory elements to genes a lot, and it's still very unsolved, I think. Could you go a little bit into more detail how H-MAGMA works? Are you assigning to all variants a regulatory element to a gene within a TAD, or are you expecting there to be a loop between the regulatory element and the
Oh,
gene?
yeah, we're only using loops here.
Okay.
So we're not really counting the TAD because sorry, I still don't know where my cursor is. But yeah, when it comes to H-MAGMA, we only use the loop information to link variants to the genes. So first of all, if we filter out any variants that are located in either exons or promoters, and promoters are just defined as 0 to 2 kb upstream of the promoter definition or the transcription start sites. And then for the variants that are mapped to those, we just assign them to those genes. So exonic variants or promoter variants are assigned to those genes.
Mhm.
It still means that 98% of the variants, or 95 to 98% of the variants, are going to be non-annotated because they're in the non-coding region of the genome. And only for those variants, we are testing how often they are interacting or in the loop with any other promoters of the gene. So we're only using promoter anchored loops here rather than just all loops. And then if those variants can be anchored back to the promoters using Hi-C data sets, then we call that those SNPs can be mapped to those genes. Does that make sense?
Yeah.
And then we just collapse all that information at the end of the day.
And what fraction of variants can you do that with?
Oh, the original paper actually has all the SNP counts, but I think it was very similar to the number of when you're just counting the number of SNPs, it was very similar to the number of SNPs that MAGMA can call when you're using like really large window. So it is not annotating every single variant within the genome, but it was still annotating a huge chunk.
Okay.
Yeah.
Thank you.
Mm-hmm.
I have a question. I was really interested in the observation about the differences between the EQTL versus GWAS SNPs. What do you think is behind that? Is it just a sensitivity problem with gene expression analysis?
So what they have suggested was evolutionary selective pressure effects. So GWAS is actually balancing the cases and controls, right? So those cases in the general population are going to be very minimal, right? But they're actually putting those into almost 50% when it comes to GWAS associations. So it means that GWAS can actually bias or make it more disproportionate when it comes to variants that are undergoing selective pressure. However, EQTLs are usually coming from the general population, and usually they're trying to generate that from healthy individuals. And because of that, the majority of the variants that are associated with expression should not undergo any selective pressure. So any variants that do not really affect any important genes are going to be first identified. Of course, if you expand the search space and if you just increase that to millions and millions of people, at the end of the day, you will be able to identify the variant gene relationship for almost everything. But at the current power, we are basically selectively more identifying the variants that are not really important for human health or disease. I think that was their interpretation. And David Goldstein's group has also shown the same thing before, that disease-associated genes or dosage-sensitive genes are more likely to have really complex regulatory relationships. And it's because if one of the enhancers are knocked out, that gene can have very deleterious effect of the human health, so those are selected out. So really important health-related genes are more likely to have multiple enhancers to buffer out their gene expression. And therefore, those variants are also going to be buffered out when it comes to EQTL study design. Does that make sense? Yeah.
Another question, it was kind of striking to me that you found about 5 SNPs from the MPRA at each locus. So were those SNPs in close proximity to each other? Like, would you consider them to be in the same regulatory element or maybe disrupting the same transcription factor?
That's actually a great idea. We haven't tested that at all. I think I definitely need to test that out. The major reason that we actually generated this was because indeed, even after performing MPRA, the majority of the loci, I think only 19 loci could be summarized into a single variant, and it's still the majority of the loci were still multivariants. So we had to come up with a plan to summarize all this, right? So that how much of the pleiotropy exists when it comes to multivariant loci, but haven't really tested that question per se, whether those variants are acting in the same regulatory element or logic. I think that's an excellent question. We can definitely ask that.
Any other questions? Any questions on Zoom?
We can't see anything.
Okay. All right, then we'll wrap up. Thanks, Hyejung, for a great talk.
Thank you, everyone.